Hi, my name is Arendt-Tranholm A. I'm a postdoc at the Center for Advanced Pain Studies, and I'm here today to talk about my paper on the RNA isoform expression landscape in the human dorsal root ganglia generated from long-read sequencing. So RNA isoforms are variants of transcripts that are generated from the same gene. This aids in generating immense complexity of the human proteome, as different proteins or variants of proteins can be generated from the same gene. RNA isoforms are generated through a process called splicing. So this is a post-transcriptional mechanism, which means that minor or sometimes major parts of the sequence within the transcripts can vary. This means that there can be changes both to the coding potential of the transcript, but also to the final protein product. So splicing has been implicated in multiple different biological processes. One of these is neuronal development, so this includes both cell differentiation, but also axonal guidance. In the last decade, we've really seen advancements within the field due to the development of advanced sequencing techniques, which means that we've been able to elucidate the complexity of the transcriptome of the human peripheral nervous system. The goal of this paper in particular was to use a new and exciting sequencing technique to characterize the isoform expression landscape, specifically in the human peripheral nervous system. So we used long-read sequencing, specifically PacBio long-read sequencing, which is a circular consensus sequencing approach, and it allows us to get full-length transcript resolution. Full-length transcript resolution is important and interesting because it allows us to look for and try to identify novel isoforms, which can contain novel combinations of known splice junctions or entirely new splice sites and splice junctions. So the aim of this paper was to look at the expression pattern of both previously annotated and novel isoforms in the human dorsal root ganglia. We then validated our findings in deeply sequenced short-read sequencing data from different human organ donors. Really interestingly, we identified 763 entirely novel isoforms, which contained at least one novel splice junction. So these are isoforms that previously have not been annotated at all. And interestingly, the majority of these isoforms were actually of genes that were most highly expressed in neurons. It was really important to us to make our findings publicly available and easily accessible. We did this by creating a highly interactive resource, which allows the user to look up their favorite gene and find the expression pattern for both previously annotated and novel isoforms that we identified in the human dorsal root ganglia. One of the findings that I was personally really excited about was the identification of multiple novel isoforms for genes that are already of interest to us in the field of chronic pain research. One of the key findings that we highlight in this paper is the discovery of multiple novel isoforms for the gene HNRNPK. HNRNPK encodes an RNA binding protein, and all 16 isoforms that we identified with long-read sequencing contained a novel elongation of an exon, as well as the excision of a different exon. We predict that this could mean the removal of a specific phosphorylation site, as well as the addition of a new one, which could have immense functional implications for the role of the protein. This was a brief summary of the research that's gone into our paper. We hope you enjoy reading it, and feel free to reach out if you have any questions.